There's some evidence for sort of anti-inflammatory effects independent of the caloric consumption, at least that's what's claimed, from GLP-1 agonists, which we would expect could have positive benefits in the context of aging.
Matt Kaeberlein, when we look at GLP-1, we look at Ozempic, semaglutide, any GLP receptor agonist, we immediately think glucose, we think weight loss.
Yeah.
But you study longevity, and I know you're not a GLP-1 scientist per se, but you have some interesting theories or ideas on how GLP-1 could potentially be impacting longevity. I want to talk about that. What have you seen? What are you curious about there?
Sure. Well, I think first I'll start with what I haven't seen, but really needs to be done are sort of the basic experiments asking whether or not GLP-1 agonists can increase lifespan and health span in animals, right? That's kind of fundamental to how I think if we're going to study aging, we need to know, does this intervention impact longevity? Those experiments haven't been done. It's kind of a missing piece in the literature. So I'm hoping that will happen soon. I think there are really a couple of ways where GLP-1 agonists are predicted to have a longevity benefit and potentially target the biology of aging. The obvious one is through caloric restriction. So we know that caloric restriction is the gold standard for interventions that increase lifespan, appear to slow aging in laboratory animals. And we know that GLP-1 agonists through appetite suppression will induce a caloric restriction state. Again, important to say, we talked about this earlier, that caloric restriction in people, chronic caloric restriction, I'd be careful about as a longevity intervention. I think there may be some consequences if you don't also pay attention to lean mass and bone density and frailty that could offset any benefits from caloric restriction. But in laboratory animals, it's a very potent way to increase lifespan. Interestingly, we also see some concerns with GLP-1 agonists when it comes to loss of lean mass, loss of bone density. So again, it kind of makes sense that this is going to line up. I think what has me most intrigued are these potential other functions of GLP-1 agonists that seem to be independent of caloric intake or appetite suppression. So we know that the receptor is present on many different cell types and in many different tissues. There's some evidence for sort of anti-inflammatory effects independent of the caloric consumption, at least that's what's claimed, from GLP-1 agonists, which we would expect could have positive benefits in the context of aging. So I'm intrigued by the possibility. I just don't know of any data yet. And I will say, I think one of the challenges is going to be, how do we really separate the appetite suppression effects from other effects of GLP-1 agonists? And so there are some experimental approaches you could take to do that, but it's non-trivial, I would say. But I'm sort of, again, in the absence of any data, I'm pretty bullish on the possibility that GLP-1 agonists could actually positively modulate the biology of aging and have effects on health span and potentially lifespan.
I put a link down below for 30% off all things from Thrive Market on your first order. Now, they're not just about snacks and pantry staples. They've got good, legit, high protein foods, which is how I live. Even my snacks are high protein. Thing is, if you go to a grocery store, the high protein snacks, they're kind of playing games with you. Like, they'll call it high protein, but you look at the ingredients and you realize that there's like some weird kind of like pea protein or something. Not that there's anything wrong with pea protein, but the reality is it's not what it seems on the surface, and it might only be 4 grams of protein. The thing I like about Thrive is you see what you get and you get what you see. It's transparent. So you can look at the ingredients and they're not gonna have garbage there, and you're gonna be able to see it. And then it gets delivered right to your doorstep. So you're not having to spend time. Even myself, I know what to look for at a grocery store, but it takes me probably 25% longer to go grocery shopping than the average person, because I do cross-reference everything that's in the ingredients. I don't like to bring that stuff into my house. Thrive Market takes that guesswork out of the equation for me, and it makes it accessible to me, but it makes it accessible to people all over the country. So that's what's really cool. So that link gets you 30% off your entire first grocery order and gets you a free $60 gift. Not to mention it gets delivered right to your doorstep, which is pretty darn cool, and their shipping is speedy, speedy fast. So that link down below.
Well, I mean, even the appetite suppression is not necessarily the intended use of GLP-1 originally, right? So it's like that kind of came as a, whoopsie, this is cool.
Yeah, that's right. Sometimes these happy accidents happen.
Sorry, I was going to make a joke about me being a happy accident person. But we'll leave that. So, we kind of pivot over to the mitochondria for a second
Sure.
and talk about that, because I think there might need to be a little bit of education as to like what the intended use of GLP-1s are, right? Because like people go and use it for appetite suppression or for weight loss, but its real effect is like for glucose homeostasis and kind of the management of diabetes, right?
Well, that's how it was first approved, right? So it was first FDA approved for diabetes. I think we're learning now, and again, this is a little bit outside my area of expertise, so I might get this wrong, but I think that the effects on glucose homeostasis still have not been clearly separated from the effects on appetite suppression, right? So that it may turn out, again, a happy accident that these drugs have a very potent effect on both obesity and diabetes and potentially kidney disease and potentially dementia because of their appetite suppression effect primarily. Again, I think we're learning that there may be effects independent from caloric consumption, but that still hasn't been completely fleshed out as far as I can tell.
Yeah, and then how do you separate caloric restriction from the actual just loss of fat mass too, right? Like, so
Right.
like these two different
Which
things.
is going to have an impact on a bunch of other things, including metabolic health.
So if we look at metabolic health, I mean, let's kind of work backwards on the mitochondria here for a second. Like if we were restricting calories, whether it be fasting, whether it be GLP-1 receptor agonists, whether it be just like grinning and bearing it through caloric restriction, like what starts to happen in the mitochondria in someone that is, let's just say, moderately metabolically dysfunctional?
Yeah. So, I mean, again, I think at the sort of general answer is things get better. And there's a lot, again, it's very complicated. It depends on the tissue you're looking in. A lot of stuff happens when we are calorically restricted that rewires metabolism. I think one of the things that often gets pointed to is an increase in autophagy, and one type of autophagy is called mitophagy, and that can turn over damaged mitochondria. So that happens and probably has a net improvement in global mitochondrial function. There's also a change in just the way that carbon is metabolized in our cells, and this is where, again, I'm struggling, as you can tell, to try to give a simple answer. The reality is it's complicated exactly what happens to mitochondria, but in general, there seems to be a boost in mitochondrial metabolism in response to caloric restriction. Rosalind Anderson, who's at the University of Wisconsin, has done a bunch of work on this where they've looked in mice on caloric restriction, done transcription, proteome, metabolome, and they see these increases in mitochondrial biogenesis and expression of mitochondrial proteins, suggesting that probably total mitochondrial capacity increases in response to caloric restriction. So at the same time that you're having a turnover of damaged mitochondria, you're getting a boost in mitochondrial capacity. And the simplistic answer, and this is where most people will gravitate to, is this is just a way to maintain your energy stores, right? To increase or to maintain the amount of ATP that's available. The idea that mitochondria are the powerhouses of the cell is true, but incomplete. Mitochondria do a whole bunch of other stuff that's really important. And so, it's probably a combination of maintaining energy stores, but also maintaining all of these other functions that mitochondria are involved in that are required for cellular health, cellular function, cell division.
So, I mean, hypothetically, if someone were to maintain their exact diet, exact level of percentage of caloric restriction throughout their life, right?
Yeah.
So would mitochondria count and quality change as we age?
Probably still change. Would it be attenuated under caloric restriction? Perhaps. I think, one of the things that we know about caloric restriction, this isn't exactly answering your question, but again, it's, I think because we don't really know the answer, but one of the things we know about caloric restriction in mice is if you go to total metabolic rate, that goes down with caloric restriction, as you would expect. But if you normalize to body weight, it actually goes up. And so it could be the case that there's actually, on a per mitochondria, and again, there's lots of assumptions there about does body weight completely correlate to mitochondrial number, but on a per mitochondria or mitochondrial mass basis, there might actually be higher metabolic activity under a caloric restriction state. And it seems to be the case, at least if you look at total body weight, that that's the case. Chronically in mice, people might be different. I don't know if those studies have been done carefully in people. There was a controlled clinical trial, actually several of them, that go by the acronym CALORIE. So these are human calorie restriction studies. And in at least a couple of them, they did use metabolic rooms where they could actually carefully measure metabolic rate and a bunch of other parameters. I don't recall that there was a strong signal on metabolic rate, but that data may be out there.
And it's absolutely okay if you don't have an answer to this one, because I'm just genuinely curious.
Okay.
So there may very well not be an answer, or at least your opinion. If calories were still maintained and someone were to take GLP-1 receptor agonists, do you think there is a benefit? Do you think anything happens?
I'm hopeful. Yeah. So I don't know the answer. I'm intrigued by the initial early data. I think there are some indications, certainly, that GLP-1 agonists have effects independent of caloric consumption. I'm hopeful that they're beneficial. I don't think we know yet.
So if you, again, I'm not going to put you in a spot where you can dangerous territory, but if you had to make a guess as to where there potentially could be that happening, under what direction, what sort of downstream effect would it be?
Yeah, again, I don't know enough about the biology of the GLP-1 receptor or what's been shown in the recent literature. I have read and heard a lot of talk around anti-inflammatory effects. So that's a catch-all term, and it's kind of like antioxidants. People overuse it. I overuse it, but that would be one of the places where I would look. I'm also intrigued by the idea that, for example, the receptor exists in the brain. Yeah. Could there be effects? And there's evidence for sure for the GLP-1 agonists for potential effects on other types of addictive behavior in people taking GLP-1 agonists. So could there be changes in brain function that are impacting behavior, mood, potentially other aspects of daily life that our brain is involved in regulating? That could be important here.
Yeah, I actually just I think released at the time of recording a video 4 or 5 days ago about interesting GLP-1 receptor agonists influencing GABA, right? So creating a significantly more GABAergic state, even though they're in a calorically restricted state, which may otherwise make you more sympathetic, right? So it's kind of interesting to see more sympathetic tone, even though there's caloric restriction. Right. And what does that do downstream of that? A lot of good things come from being more parasympathetic, right? So, and I don't know, I just find it really fascinating. And if we go into your area of research where you're really strong, is particularly in the world of rapamycin and stuff, and I think that's how we got on this topic originally offline when we weren't recording. Is talking about how there's an impact with rapamycin and people saying, "Hey, my appetite is suppressed," and maybe there's something going on there with GLP-1s and how that's all.
I agree. I mean, this is something I've thought about and never done, but it would be really interesting to know in people taking rapamycin, is there a change in GLP-1 expression level or receptor signaling? Doable experiment. I just don't, maybe there's data out there. I don't know of any data.
Yeah. So if we jump back again, mitochondria for a minute, what would you say, and I know we're kind of bouncing around a little bit, but when we talk about caloric restriction, I think the basics that people can grasp are okay, well, I'm metabolically going to be healthier, right? So we talked a little bit about what happens when you restrict calories, what may be happening with the mitochondria. Is there a stress to the mitochondria when you are restricting calories to the point where the adaptation is to get more efficient? Mm-hmm. And that's kind of one theory that I think surface level, in which case, what does that do for performance? What does that do for ATP capacity? What does that do for fat loss, even fatty acid oxidation?
Right. Changes in substrate availability and metabolism,
Exactly. So, and if we were to look at that, I kind of want to take independent of caloric restriction, what can people do to improve mitochondrial health?
Right. I think the most obvious thing is exercise. That and even more so than caloric restriction. I think it's clear that regular exercise has benefits for mitochondrial function and mitochondrial health. So that would be the first place I would think of, even above caloric restriction, in my view.
Yeah. So if we're looking at more mitochondrial density, which is essentially just more mitochondria in a given area, is there, and might be a complicated question, but at the same time very simple. If there's more mitochondria in a given area, obviously that's more mitochondrial density, more ability to create energy in a given area. Can you increase the efficiency of one individual mitochondria, or is it really more about just getting more density and more of it?
And I would even say, density being more mitochondrial capacity assumes the mitochondria are working, right? So you can get more density if you're blocked for degradation of mitochondria, right? If mitophagy is inhibited. So, but making the assumption that they're functional mitochondria and they're doing their job. So again, I feel like this is an area where the perception in the scientific circles continues to shift. Like, is it the case that it's discrete mitochondria or this sort of overall network that's most important? And even the way we measure mitochondrial density, if you think about how that's actually measured, it's either usually the number of mitochondrial genomes that are present or the amount of mitochondrial proteins that are mostly or completely localized to the mitochondria. So is that a perfect measure of mitochondrial density, especially since we know that mitochondria exist in this dynamic system where sometimes they're interconnected, they share membranes with each other, and sometimes they're distinct, right? So fusion and fission of distinct mitochondria. And we're learning, sorry to make it more complicated, but we're learning that the mitochondrial membranes are even sometimes shared with other membranes in the cell, like the endoplasmic reticulum. And those connection points are really important, people are learning. So I don't know that it's necessarily going to be about density per se, or even whether they're fused or fizzed, right? Whether they're distinct mitochondria or in this sort of interconnected network. I think there's just a lot that we still don't know. I think in general, some of the interventions like exercise, like caloric restriction, have a big impact on the mitochondrial network. And for sure, are changing metabolic rate, and as we've sort of alluded to, substrate availability, substrate utilization, ATP levels, NAD homeostasis, all of those things are interconnected. And in general, when people are healthier, their mitochondria are functioning better. And I think there's reason to believe that there's a causal arrow there, right? That better mitochondrial function is going to lead to improved health in most cases. And the reverse, impaired mitochondrial function leads to disease in many cases. And we haven't talked about this, but some of the work that we did in my lab years ago now was looking at what happens when we have very severe mitochondrial dysfunction. So like kids who have these inherited mitochondrial disorders, and how that intersects with nutrient signaling and caloric restriction. And to our surprise, what we found was that you could actually rescue some of these severe mitochondrial defects in different models using caloric restriction or mTOR inhibition, which was counterintuitive at the time. I think now that we know a little bit more about the network makes sense. But back then we didn't expect that. And when we started publishing these papers, I think a lot of people were surprised by it.
Yeah. And you said something that I'm hung up on for just a minute. So you can basically have essentially dysfunctional mitochondria that can replicate too, right? So that mitochondrial DNA is still, so that's something that once you learn, you kind of can't unlearn, because it's just kind of scary. So if I have a bunch of dysfunctional mitochondria and I'm increasing density, but I guess at the same time, if you're doing the right things to improve mitochondrial health, it's not like you're just going to replicate a bunch of dysfunctional mitochondria, right? If you're exercising, then you start to improve. So I don't want people to think that like, okay, I've got bad mitochondria, it's only going to replicate a bunch of bad mitochondria.
Yeah, and there's a few things to say about that. I mean, I think, first of all, many people may not appreciate this, but even when we talk about discrete mitochondria, right, as a unit, there are hundreds or thousands of genomes in that unit. And so there's actually mixtures of mitochondria with different mutations within one mitochondria when they're in this distinct unit. And so what can happen is there are sometimes mutations that are preferentially replicated. They can replicate faster. They can divide faster than other genomes, and they'll start to take over. They'll become more dominant in that discrete unit. I think one of the reasons why mitochondria go into these discrete units is because there are sensing mechanisms, we think, that can tell how well that discrete unit is functioning. And when it's not functioning well, you have proteins that come bind to the surface of that dysfunctional mitochondria, and that targets that mitochondria for degradation through autophagy. So our cells have evolved actually ways to sense when one of these units has a preponderance of damaged genomes or for other reasons isn't functioning well, and we target that for degradation. And there are diseases where we know that particular pathway that targets mitochondria for degradation isn't working well. Things like Parkinson's disease can be caused by an inability of the cell to appropriately recognize which mitochondria are damaged.
That's pretty interesting. Yeah, it's almost as though mitochondria are a little bit more than a powerhouse.
They're a lot more.
So I kind of want to go back to, so you look at disorders, or in children when you see like mitochondria already having this dysfunction. Are we starting to see now? I've seen some early literature that like this is something that actually can be passed on, right? So like we're seeing metabolic dysfunction starting to get passed on into pediatrics, which is a little bit scary.
So there are two ways that can at least two ways that can happen. So there are mutations in our genomes, and they can be in and if you think about a cell, there are two types of genomes. There's the nuclear genome. That's what most people think of when they're talking. Well, if you go to 23andMe and get your genome sequence, that's what you get sequenced. Then there's the mitochondria genome, which are distinct from the nuclear genome. You can have mutations in either of those that lead to mitochondrial dysfunction. I should know the exact number, but it's like 90+% of the proteins in the mitochondria come from the nuclear genome. They're encoded by the nuclear genome. So it's a tiny fraction, probably more than 90% now that I think about it, that actually come from the mitochondrial genome. But the ones that are in the mitochondrial genome are really, really important. So when you have mutations that cause a loss of function in those mitochondrial encoded genes, that usually will lead to a severe disease. You can get less severe diseases when you have mutations in the nuclear encoded mitochondrial. So those would be things that maybe you don't notice at all, but you're metabolically impaired because of a genetic mutation, or you don't notice it till you're 50 or 60 or 70. And each of those can get passed on. The severe ones, oftentimes, you're going to know if you're a carrier, for example, because somebody in your family has had it, and there are tests that can be done for a lot of these. A lot of the ones that are nuclear encoded, we're still finding. Like there are lots and lots of diseases that are caused by mitochondrial defects that haven't yet been mapped to the mitochondria. And certainly, and I'm not trying to scare people, this is just a fact of genetics. You could be a carrier, your spouse could be a carrier. You get those two together, and that's when it becomes a problem, when it gets passed to your kids. That's rare. These severe mitochondrial diseases are extremely rare. So again, I'm not trying to scare anybody. And I think we're now starting to have tests where we can see in adults if they have impaired mitochondria. And again, some of these are going to show up as, impaired glucose homeostasis, predisposition to diabetes, things like that. The other way, though, that mitochondrial dysfunction can be passed on is through epigenetic regulation. So we've learned now, this is the idea that there are changes on top of the genome that control which genes get turned up or turned down. Methylation is one of the big ones that people talk about in the aging field. So these epigenetic changes, which can be induced by your environment, how much pollution you're exposed to, what foods you're eating, famine, that will change your epigenome. It'll change which genes are turned up and turned down, and sometimes that can be inherited. And so all sorts of traits, not just mitochondrial dysfunction, all sorts of traits can get passed on epigenetically. And this is where, there's a lot of interesting study around how does the maternal environment affect the expression in the progeny in kids. And we know a lot of things that we control in the maternal environment, and we're starting to learn about more things we probably should be thoughtful about.
How long does it take for mitochondria to essentially turn over? It's
I don't know the answer.
Yeah, because it seems like from a metabolic perspective, that's a lever that people can at least have a little bit of control over, right?
Here's what I would say. I think it's going to be very dependent on what you're doing, right? So things like diet, exercise will change the rate at which mitochondria are turned over quite dramatically. Again, we've talked about autophagy already. Autophagy is sensitive to nutrition, so fasting will boost autophagy. In most cases, that will lead to an increase in mitochondrial turnover. Same thing with exercise. So it's going to depend a lot on your lifestyle, your activities, as well as your genetic predisposition.
So, in theory, though, each time you're kind of turning over, that 7% that you can control in terms of like the mitochondrial DNA.
Yeah.
Do you stand a better chance at kind of improving that?
Well, and I would say it's much more than that 7%, because if you think about the way the mitochondria are functioning, again, this will go back to sort of foundational biology, right? You have your DNA in your genome that makes a RNA, which makes a protein, and this protein is the machine that does the job. Those proteins go through the cytoplasm into the mitochondria, get imported into the mitochondria. So they're in that unit. So even if they're dysfunctional to start with, it's not going to completely impair the function of the mitochondria right away. It's going to be a combination of maybe a slightly dysfunctional nuclear encoded protein with time that leads those mitochondria to wear out faster. So it's not only the mitochondrial mutations that can benefit from turning over these damaged mitochondria. It's a combination of where the mitochondria start. This is a little simplistic, but where they start functionally, and then how fast that function declines over time. That's going to lead to there's some threshold then when the cell is going to detect it as a problem and degrade that mitochondria.
Interesting. So if we come back to rapamycin again, is there some speculation that could actually increase the mitochondrial turnover then?
It almost certainly does, at least initially. So again, when you inhibit mTOR, you're going to boost autophagy. Part of that is going to be boosting mitophagy. Part of that's going to be at least a transient increase in mitochondrial turnover. Whether that's why or related to how rapamycin seems to improve severe mitochondrial dysfunction, still a pretty open question. We don't really know. And again, that was a blanket statement. It improves severe mitochondrial function for certain types of mutations. We don't know how broadly true that is. We know it's true in mice, some case reports in people, but still being worked out.
But it's interesting when we have this discussion and we bring it back there, we can close the loop on saying like, that's exactly why there could be a potential link with GLP-1s and also possibly even the co-administration of GLP-1 and rapamycin.
For sure, right? Yeah. And again, that's a really interesting thing I've thought about. I don't know of any data. I'm sure there are biohackers out there using both rapamycin off-label and GLP-1. I haven't seen data from those people. It's really intriguing to think about what might happen.
Yeah, no, and I encourage people to look at GLP-1 with a grain of salt, right? It's not all about just the Hollywood Ozempic craze. There's people that are really getting benefit out of it,
yeah.
And there's also promise there, and that we shouldn't be so quick to just shoot something down because Hollywood's using it.
Oh yeah, you know? and I would say again, I think this is probably most people have heard this by now, but it's worth saying. I think the way I sort of view GLP-1 agonists is they are a really useful tool to help people get started on the path towards improved health, especially if you're obese, you're diabetic, you've struggled with body composition and body weight. They're not going to be a cure-all, but I think for people who've struggled, it can really help people get on that path, start to see improvement. We know that there are impacts on behavior, which seem to be positive from a food consumption perspective. So I think they can be really valuable. And we've learned a lot more now, but at least when they were initially rolled out, they were not paired well with education around what you're eating and the importance of maintaining muscle mass and doing resistance training. And I think that's really important for people to understand. Yes, you won't want to eat as much, but use this as an opportunity to improve your diet, right? And that's what I mean by it can be a really useful tool to get on the right track. And definitely pay attention to your muscle mass and make sure you're doing resistance training. I personally think if you're on a GLP-1 agonist, you should get a DEXA and watch your muscle mass, watch your bone density, and make sure you're taking steps to maintain that.
Yeah, that's the big one I was going to mention is the bone density. I mean, you're talking about older populations that are using this that are already at risk. And I think that initially it was brought on as this lose weight without doing anything sort of appeal. Like, you don't have to diet, don't have to exercise, you're just going to lose weight because you're going to eat less. I think now it's come under enough scrutiny that I think people know, okay, hey, like prioritize protein, prioritize resistance training,
I'm not sure. I think people who listen to your podcast know. I'm not sure that's true for the general public. And I know a lot of primary care docs still don't emphasize that. So I think the message still needs to get out there.
Well, that's why we're doing this. Well, Matt, where can everyone find you and where can everyone find Optispan?
Yeah, so Optispan website is www.optispan.life. And our podcast, the Optispan podcast, is on YouTube, and I definitely encourage people to check it out.
Right on, my man. Thank
you.